All right, here we go again. It is Saturday, January 29th. We're doing part 2 of the Managing Microbes Masterclass. We got some homies in the chat. Thanks for showing up early. Did it out of order. I guess we got to hit the first. And yeah, what's up? We're about to do some science. Hope you're stoned. If not, fix it. So yeah, man. So I guess, what do you think? Just jump right into it, or?
Absolutely.
All right, here we go. All right.
Okay, so this masterclass is going to extend a little bit on what we've been doing in the previous 2 episodes of this masterclass series. This one will specifically be wrapping up the case study we've introduced to you on the managing of the microbe component of that case study. So let's jump right into it. But first, upcoming live events. What do we got going on?
Tomorrow we're going to do part 2 of the Soil Balancing Masterclass. I'm going to cover some commonly found problems that keep people from sorting out the math. So we're going to look at some common examples that we run into that makes it really confusing from a math standpoint.
Yeah.
And then again on Wednesday, February 2nd, we're going to be doing episode 2 of Science Bro, where we're going to be breaking down some science and hopefully bringing everybody up to speed.
All right, so managing microbes. Like I said, this is the last part of the story in that case study, and we're going to kind of dive into these components here. So microbial considerations are largely misunderstood, misrepresented, or disregarded altogether in most agricultural settings, and if you're not measuring them in a way where you can see all of the microbes you're trying to manage, then you'll miss a lot of the soil food web that could be working for you.
Which is kind of crazy. From a living soil community standpoint, we have a whole bunch of people talking about microbes, and there's just a very small handful of people that are actually measuring these things. And one of the things that's important to us is that we get some real proper and scientifically correct narratives into the stream of consciousness of the farmers out there. Because a lot of what we do up to this point is fairly incorrect from a biological standpoint. And that's not from an opinion standpoint, that's from generally accepted scientific principles that are defined, directed, and created by Dr. Elaine Ingham as far back as 1985. Which we covered in Science Bro on last Wednesday. If you're tuning in on the replay, you might want to check out that if you haven't seen that episode already. And we go over to the foundation of what has been known for how long.
Yeah.
And it's been known for quite a while. And Sarah and I have both worked very closely with Dr. Elaine Ingham. We've worked in her courses. We've been mentors. Well, I should say I had one mentorship student. I wasn't very good at it. Sarah had a lot of mentors. We'll just clear that up. But nonetheless, we've worked very closely with Dr. Elaine Ingham and have taken her work, incorporated it into our consulting work, and I feel in a lot of ways have taken her work to the next step in the commercial cannabis space.
Yeah. Yeah, we have noticed that there could be, just to get a higher health level of what's going on, because the microbes can bring you a lot of health, but there is a whole story behind managing these microbes and getting this robust living soil system that we're after. That's first and foremost. So how do we manage the terrain? How do we set the stage to allow these microbes to do what we say they're doing, right? So getting them functioning, getting them stimulated, providing nutrients all the time to your plants. And how do we do that with terrain management and understanding all the components that go into that.
So the first goal is to get that robust living soil.
Yeah, and that takes into consideration what it takes for these microbes to establish, proliferate, and continue to proliferate throughout what your growing season is. So that means you have to understand what they need. What is the food sources that they need? How do they need the water to be? Does that match what you think the water needs to be? Those are the things that you need to be able to manage when you're managing microbes. And as we're going to see throughout this, you can't manage them if you're not measuring them. And so these microbes, the plant will feed the microbes in the soil, and the soil will feed the plant. And so it's this loop that goes back and forth, this interchange, this communication that happens. And we'll see that as we go forward. This Goldilocks principle is something that we often will talk about, because when it comes to managing microbes, there's always going to be an ebb and flow in the microbial population. So an ebb and flow in your bacteria as they get grazed on, and then as they reproduce with their life cycle of every 20 minutes. Always going to be an ebb and flow. So everything that you do in the space, all the minerals that you add, all the nutrients that you add, how does that affect their ebb and flow? And if you give too little nutrients, too little moisture, how does that affect them? Or too much? Right? So we're really looking for that just right, that optimal balance in the system to really allow them to get a nice cruising speed of ebb and flow, and not these huge booms and busts that we talk about.
Mm-hmm. And ultimately, we're after balance.
Yes, balance in the system, balance in all three of these components that we're talking about. Mm-hmm. So the three components are, again, the microbes, which are the foundational cornerstone of everything that we do. They are the ones who speak to us, tell us what we're doing above ground is actually making them happy and allowing them to provide what they need to provide to your plants, or is it not? And then the other two components are the minerals, which are also a food resource for your microbes. They need these mineral components in order to do their functions, to make their enzymes. And then the nutritional component is also the way that we feed them with different food resources, the way that we feed the plant as well, all in balance together, everything working in harmony so that nothing is too much or too little, and we can get the outcome that we want with living soil. And so when it comes to this balance, there's a few key components in achieving it. What we want is we want to establish the balance of those three things, the microbes, the minerals, and the nutrients, before we start turning up the gas. We want to be able to get that ball spinning on our finger before we start making it go faster, basically. And then diversity over singularity, that's the next key component. And we're talking about diversity in all three components. Diversity in our microbes, we want a diversity in our bacteria, diversity of all of the different organism groups. The more, the merrier. They all have their functions, they're all important, and we need them there working for us. But we're also talking about diversity in our mineral components. So we don't just want nitrogen, phosphorus, potassium. We want all of them, including our trace minerals, and we want them in balance, something that will lend itself to a happy, healthy plant and a happy, healthy soil food web. So, and again, in our nutrient component, which we're not going to go over in this class, but you will get it if you take the master class series. So that diversity in our nutrition as well. Mm-hmm. And then when everything is in balance, then we can increase the intensity. We don't want to blow the gaskets off the engine first. We want to build the engine first and then rev it. Yeah.
Exactly.
All right, so microbe-supported plants. So this is hopefully not new to you, but microbes, beneficial microbes in nature improve the health of organisms that they inhabit. That's what they do. They do it in you, and they do it for your plants as well. So they increase the plant health. You got to remember, they're working at a very minute level. They're working at the genetic level. They're working with these elements, and they're speaking to your plant at that genetic level. They're giving the plant what it needs. The plant is telling it what it needs. It's going and getting it, sequestering it, and giving it to the plant in the forms that they need in order to be efficient in consuming energy and producing energy. So that production in energy allows it to store fats, allows it to produce more yield, allows it to produce higher level compounds. So like your terpene and profile, your terpene, polyphenols, cannabinoid profile come largely in effect to this microbial mineral plant interplay, feeding the soil, feeding the plant and getting all of those nutrients that they need in the forms that they need. It allows the plant to create these secondary plant metabolites. Those metabolites increase and improve the immunity to pests and disease. And we have seen that happen. We have moved a farm from pests and disease out of pests and disease, definitely by the second harvest, but we've even seen it happen in the first harvest too. So
it's pretty common that we're able to get rid of all powdery mildew within the first harvest of working with a farm simply by balancing the biological considerations we're going to talk about today.
Yeah.
If we're not able to move a farm out of powdery mildew, it's almost always because they're having a hard time stopping IPM protocol.
Yeah.
And the IPM products are oftentimes very antimicrobial, and so you can't get over the hump of protecting against pests and disease from a biological standpoint until you stop killing the organisms in the soil with your preferred IPM products.
That's right. All right, so we're just going to kind of jump into the basics of these organisms that we're talking about. So we categorize them into basically two simple functional groups. This first one is the nutrient storage. So these are your what I call cornerstone species. So bacteria and fungi, these are the ones that are sequestering all the nutrients into their bodies and building that soil structure at the micro and macro level. They're building that pore structure. Remember that fungi in your soil, they need oxygen, they need moisture. So they're, they're gluing onto the different organic matter in the soil, and they're making pore structure so that they can get that air into the soil, so they can get that moisture that they need in order to proliferate. So this is your storage organisms. So getting the money into the bank. Now we need to get the money out of the bank and into the plant, right? So these are your nutrient cycling organisms. These are all your beneficials. Your flagellates and amoeba are part of your protozoan category. So they'll have a preferred food resource of your bacteria, typically, and then also your beneficial nematodes, and they can be bacterial feeding, fungal feeding, or predatory. And whatever they eat, they'll release any excess nutrients back out into the soil. That will then become available to your plant in the forms that they need it, in organic forms. So that's the gist of what we're talking about: banking it and building soil, and then getting the nutrients out of them back into your plants. So the process looks like this: you've got a plant, you've got sunlight, the energy's coming into your plant's photosynthesizing and making energy and putting that energy down into the soil, pushing out root exudates specific to what it needs, so stimulating these guys, your microbes in the soil, to go sequester and get what they need, and then they'll go get what they need, and in that cycle they'll feed it back to your plants. It'll become available. Your plant will then bank that energy into what it needs, and then request the next set of nutrients that it needs through those different sugars. And again, the cycle happens again, where they'll bring that food back to the plant, and so on and so forth. And this is what Dr. Elaine speaks about as the cakes and cookies process, where we're feeding each other back and forth in a mutualistic fashion.
So most farmers are pretty familiar with pH, and it's on a logarithmic scale. So each time you go up in one number, it's actually many times greater, which I don't think most people know. But nonetheless, most farmers are aware that there's a goal of pH, whether pH in the soil, pH in your nutrient solutions. And in the context of rock wool or cocoa coir or pure hydroponic media, if you vary slightly from this ideal pH, there's very clear indications that the plants are not happy. In often extreme cases, you can actually lock out plants altogether and kill them. So there's this mid-range goal between acid and alkaline that most people would agree is somewhere around 6.4. The main key to having success in living soil systems is understanding that it's operating off of a different paradigm that is not necessarily pH driven or pH focused, but then has downstream effects on soil pH and root zone pH. And so what you have to wrap your mind around is that instead of focusing on a certain pH target, instead you should be focusing on aerobic versus anaerobic. Most farmers are farming plants that are what are called obligate aerobes. They rely on aerobic organisms to perform their functions. In certain wetland species or plants that you would find growing in a pond, those would be not the same as what most farmers are cultivating. And in that case, they would work with organisms that are more suited for decreased oxygen conditions. So for almost every situation, if you're a farmer growing a crop, you are working with an obligate aerobe plant that relies on its functions to be served with organism-to-organism interactions within the aerobic community. And so aerobic simply means high oxygen, anaerobic means low oxygen. And it's not necessarily a short spike where it becomes one or the other. And so you actually have a mid-range that we call the facultative anaerobic zone, which is very clearly defined in parts per million of oxygen, which has been defined in the scientific research, which is referenced by Dr. Elaine Ingham's work. So we can interpret oxygen concentrations in the zone by measuring and counting the organisms that are present, because the disease-causing organisms don't even function above 6 parts per million of oxygen. The disease-causing organisms tend to show up once you drop below 6 parts per million and you fall into that facultative anaerobic zone. Once you drop below 4 parts per million of oxygen, that's where stuff really starts to get out of hand with root zone pathogens, sudden plant death, things like that. So in 100% of the situations that we've measured, when there are disease conditions or extreme pest problems, there are very clear indicators as defined on the light microscope methods by Dr. Elaine for measuring disease-causing anaerobic organisms. And interestingly enough, one thing that I really took from Dr. Elaine that was some of the most important bits of information she ever shared to me in regards to asking about pests. And she said something to the effect of, you need to change the way you look at everything in nature. And you need to understand that everything that is present is perfectly performing its given function in nature for the context and content of that situation. One of those things that is always perfectly performing its function of indicating to you, the farmer, that you've dropped into that facultative anaerobic or decreasing oxygen zone are black fungus gnats. Those are, in my opinion, the most accurate indication that you've fallen into this range. And in other presentations, I've mentioned their life cycle, and that when you notice them, typically what we have farmers do is, if you're meticulously writing down what you're doing on a paper calendar, from watering dates to watering volumes, and say you come in on Thursday and you notice some gnats, it takes 3 to 7 days for those gnats to go from the not visible phase to the flying visible phase once they've gone through the larval stage. And you can look back on the calendar and say, oh man, 4 days ago I increased my watering volume. That is clearly a component of the gnats that I'm seeing today. When I move into next week, I'm going to adjust my watering volume accordingly. And people have a hard time wrapping their mind around this, because most farmers are in the indication that, or under the mindset that literally anything that they don't want, they need to kill. In most situations where we've worked with farmers where they take a strategy to kill the gnats, pretty much every time you negatively impact the soil food web organisms, making things worse. So pretty much the worst thing you can do is try to kill the gnats with something like Natrol, which is simply a bacteria product, but 100% of the time farms over apply the application rates of that bacteria. And whenever you introduce a large volume of any type of organism, you throw everything out of whack and you actually collapse the soil food web, which is another concept that's really hard to wrap your mind around unless you've been measuring these things. So first of all, we're not going to ever take an action to kill the gnats aside from putting in the appropriate predatory nematodes like HB or SF or SC, something like that. We're never going to try and kill the gnats. Instead, we're going to pay attention to our watering volumes, the thickness of our mulch layer, and things like that that would lead to anaerobic conditions at that soil surface.
Yeah.
The gnats are perfectly performing their function in nature by feeding on the anaerobic bacteria that are present below 6 parts per million of oxygen.
Yeah.
And if we take that one step further, again, 100% of the time, above gnats is healthy plants. Gnats and below is where you move into plant health problems. And if you have a functional understanding of how this soil food web system works, it can be pretty clear to you.
Yeah.
It's not clear to the people that aren't measuring things, and this whole concept is one of the things that, interestingly enough, we get the most pushback on, but from my view of the analysis, this is the most accurate consideration we've provided to the community. So we've color-coded these bars to match the graphics that Sarah's created for interpreting and presenting our soil food web data, and you can see that from green and blue boxes, you're above gnats and you're into that aerobic zone, and the orange and red boxes means you're in an undesirable state, likely an anaerobic indicator. When we've gone through the literal hundreds of farms of analysis that we've done from a biological consideration, one of the most consistent phenomenon we see is that when there's thrips present, you are most often still in the aerobic side, but extremely low populations. Most often, those really low biological, I just got to pause to hit this comment real quick. That's what's up, Mr. Cub. Thanks for paying attention. Yeah. What up? Let us know in the chat how the plant growth has changed and the results as a result of making the change to the gnats. So most often in living soil systems, and cannabis farmers in particular, when they're battling thrips, this is one that you're actually creating in a certain sense. My personal interpretation of thrips is thrips are perfectly performing their function in nature when the nutrient cycling organisms are too low, leading to too slow of delivery of nutrients to the plant that is relying on organism-to-organism, predator-to-prey interactions. Most often, these biological populations are too low and below what Dr. Elaine Ingham describes as the minimum threshold for populations of bacteria and fungi in a soil system. Pretty much 100% of the time.
Yeah.
I would be so bold to say 100% of the time, but you have to be careful in science of making very bold claims. But
that's the trend that we see in the pattern recognition.
I would stand behind that one. I'm pretty good at, and most often the steps to kill the thrips, most often in cannabis, is neem oil spray. Does a great job at protecting the leaves from thrips. It does break the life cycle and reduce them temporarily, but will undeniably keep you in that low biological population range where you are in the desirable range for thrips.
Where you always need the neem.
Yeah.
Yeah, we're trying to fix the cause so you don't need it going down the road.
Yeah, exactly. Yeah. And once you start to get in high green boxes and into the blues, this is where we started to see over 30% total cannabinoids. And this has been a while now. This was back in like 2016 where we first created this graph and considerations. But when we were working with commercial farms that were doing a tremendous amount of potency testing, we noticed that when we got them out of thrips and up into all green and blue boxes, they started seeing some really remarkable things happen with potency of the same exact genetics in the same exact cultivation space with relatively the same exact process, and the only change was moving up into those higher desirable ranges of beneficial aerobic organisms.
So again, talking about how these microbes, when you get them functioning and in these zones that he's talking about, now you're creating your polyphenols, now you're creating your terpenes and your cannabinoids at the levels that we want to see them at, that protect the plant. Again, all of these compounds protect the plant.
Mm-hmm.
So.
And we found that once you get above the green even, so green would be acceptable, desirable, positive ranges, blue would be in those higher, and now you're starting to compete with hydroponic growth rates. Mm-hmm. I like to relate it to undercurrent systems. If you get a living soil system with that perfect, consistent moisture through like a blue mat system or just a really good watering practices, you will have very fast nutrient cycling and extremely remarkable plant growth rates. This is often what people just define as fish stories, where they just flat out don't believe us. And now if you continue to go more anaerobic past gnats, typically the first thing you see is spider mites. So in spider mites, you're going to start seeing an excess of nitrate nitrogen, which is coming from the excess of ciliates, which is an anaerobic predator of bacteria that releases nitrogen in the form of nitrate. This is fine for vegetative growth, but it is not good for reproductive or flowering growth. If you go to the hydroponic store and look at any set of nutrients and turn around the grow and bloom formulas, most often there's a larger percentage of nitrate in the veg and a lower percentage of nitrate in the bloom with some ammoniacal or sometimes urea nitrogen. And from a biological standpoint, when you are anaerobic and you're relying on ciliates and omycetes to do nutrient cycling, they are predominantly producing nitrate, which is not that desirable for reproductive stages of flowering plants. When you get into that biological range, even if your environment is fine, even if your nutrient balance is fine, there's a disruption in the types and amounts of nitrogens in the soil that are incorrect for the stage of plant that is flowering, and you get spider mites. If you go even more anaerobic than that, then you start to run into the really problematic issues, which is russet and broad mites and root aphids. 100% of the time, when farms had root aphids, they were also quite anaerobic, most often to tea brewing practices or some other really problematic biological activities. A lot of KNF deals with root aphids, russet mites, and broad. That's because they are relying 100% on anaerobic disease-causing organisms. So this is a scale that has been very consistent in our work and all the analysis that we've done.
All right, so we're jumping over to that case study that we've been talking about in the previous few episodes. So again, we came into this particular project at week 1 of flower, and this is what they looked like on the picture on the left. They had 10,000 square feet of canopy. Greenhouses 1 through 4 had remineralized soil from previous years that had 3 times the desirable nitrogen in the soil. And then greenhouses 5 through 7 had half the desirable calcium levels. So 2 different soils and 2 different issues with the mineral component in those soils. The soil was also anaerobic from this tea brewing practices. They had taken some advice on tea brewing that was not the most beneficial advice for the crew, but the crew was very solid. They were exactly implementing what they were taught. It's just that what they were taught led to some undesirable results. So this is the breakdown of what we found. So mild to moderate mite pressure across all the greenhouses.
When we started.
Mm-hmm. Oh yeah, not the results. The results of the first site visit when we were initially getting there, sorry. Mm-hmm. So mild to moderate mite pressure across all the greenhouses. Greenhouses 1 through 4 had the highest mite pressure because of the excessive nitrogen and the tea brewing process. So again, all that nitrogen in the nitrate form, because they're anaerobic, being absorbed by the plant, causing excess nitrogen in the plant and summoning basically the pests. And then greenhouses 5 through 7 had moderate mites and mild powdery mildew because of the low calcium levels and, again, the tea brewing practices that were implemented there as well.
So it's important to understand that there's biological factors that drive pest and disease manifestations, and then there's also soil chemistry or nutrient balance phenomenon that lead to above ground pest and disease issues. In this case, greenhouse 5 through 7 had very low calcium levels, and the liquid fertilizer strategies that they were taking were maybe 2 to 3 times high in nitrogen as well. So when you have a deficit in calcium and an excess of nitrogen, That is almost 100% the formula for powdery mildew from a chemistry standpoint.
Right. Yeah, and then you add on to that the soil food web component to that as well.
Yeah,
And
so
it's
they were
perfect.
getting powdery mildew factors driven from soil chemistry, fertilizer strategies, and the tea brewing practices.
Yeah. So the crew was spraying IPM products 3 times a week for the control at the start. And then by harvest time, mites were only in one corner of greenhouse 2. And then by harvest time, the PM was gone until, if you've been following along, there was a massive storm that hit that blew off their they had an outside dep.
Yeah. But it flooded the soil, and anytime there's excessive moisture in the soil, regardless of how good the biological populations were before that flood, from the point of the flood until the flood backs off, you're in that anaerobic, disease-causing, powdery mildew-causing scenario. And in living soil especially, pretty much the main cause for powdery mildew issues is people don't realize they're actually overwatering. It might not seem like you're overwatering, but from a biological standpoint and from a plant needs standpoint, it is excessive watering.
Yes.
And once that soil gets to the excessive watered state, you're now by default decreasing oxygen. All the beneficial fungi that are good for obligate aerobe plants and higher level functions start to go to sleep or dormant and eventually dry off or die off. And those powdery mildew causing, disease causing omycetes start to function because they're more suited to those standing water situations.
Low oxygen
From
situations.
a casual definition, the organisms that we measure in the soil that are very directly causing and correlated to powdery mildew issues are more suited to water molds.
Yeah. Okay, so let's just jump right into the bioassays themselves, and we're going to track 2 different greenhouses. This first one is greenhouse 1. So when we started, we took we started in August, week 1 of flower, and we took this sample on August 12th, right? So we're just going to follow this all the way down. First, I'm going to introduce you to the bioassay itself. So I've broken down the nutrient storage organisms and lumped them all in the top portion there. And underneath that is the nutrient cycling organisms. And then underneath that is the anaerobic organisms. And so if we and then you can see here we've got all the color coding that we've introduced you to on that scale of anaerobic and aerobic. So if we scroll back up to the soil building nutrient storage organisms, under August 12th, the starting numbers, we were pretty high in that. So we were higher than we'd like to see in the bacterial populations. We were really low in our total fungi populations, so there's not going to be pore space being made. They're relying solely on any aeration that either is in the soil itself. Basically, is what they're relying on for the aeration there. And then on the nutrient cycling organisms, your flagellates and amoeba are lower than we like to see it, just under the minimum threshold. They do have some bacterial feeding nematodes, so they do have some nutrient cycling going on, but at week 1 in flower, that's not going to be enough. And then when we scroll down to the anaerobic organisms, we see we're not overwatering here. We don't have root feeding nematodes and ciliates showing up, but we do have some omycetes showing up. Those aren't a ton as far as biomass goes, but they're almost double. In fact, I think they are more than double. Yeah, more than double the beneficial fungi numbers. So they're not proliferating, but they're winning as far as who's winning in the battle of omycetes to fungi in competition.
And one of the most consistent factors for visible powdery mildew on the above ground biomass is the relationship between what we define as total fungi,
which is your beneficials,
and what we define as omycetes, which are those decreased oxygen, less than beneficial to directly disease-causing organisms.
Yes. And so we adjusted the tea practices. We got rid of all of the things that led to increased omycete pressure. We switched them over to doing extracts, so they're still getting the biology into the system, but now they're minimizing any of the omycetes going in. And so we're able to and also at this point between the first and the second sample, we had done a mineral application as well. And we adjusted their nutrients too.
I think it's important to note that between the first and second sample, they're still spraying IPM products that are extremely antimicrobial.
That's right.
3 times a week. And between August 12th and 9/15, we typically can measure with the microscope within 7 to 10 days of making a single compost extract a considerable change in these organisms. And you can see in a 1-month span, there's very little change in these numbers.
That's right.
Which these should have jumped to blue. And this is oftentimes how we know a farm is still spraying IPM. In this situation, they weren't hiding it. It's just the psychology of a farmer with a lot going on. It's for those of you that are moving to Soil Food Web Advisors, 100% of the time that farmer is going to continue on with those IPM strategies because pretty much nobody before we've come in to work with them has seen a situation where a pest problem got less going into the reproductive or flowering stage. It almost always gets worse. And sometimes it can be so out of hand that the farmer loses the crop. So there's a very legitimate, reasonable thought pattern based on prior experience from the farmer during this situation. But in all situations where a farmer is able to trust that the IPM products is actually most of the problem, and they can allow the establishment of these organisms to actually take place, you can navigate right out of those pest and disease issues. And 100% of the time that the farm trusts this process and implements the analysis process of Dr. Elaine Ingham is using true biologically complete compost and operating in a way that facilitates and supports the establishment, it always changes for the better.
That's right.
At minimum, the pest pressure reduces considerably.
Yeah.
So from August to the end of September, they were still spraying 3 times a week. I believe towards the end of September, they backed it off to 1 day a week, but then they went to running Sufoil, which works well, but is also, you can see, we got them to stop the 3 things they were doing from here to here, but then, and got a slight rise in fungi, but then jumping to the Sufoil killed the fungi right away.
Right. We were able to change up a little bit of their watering practices as well, though. Remember, this was more like the heat of summer.
Sorry
and
about
so, this was like the heat of summer, and so you need to make sure that you're watering enough so that the microbes can start to increase in numbers, like we're seeing here on this particular date in the samples. So we saw that the fungi were coming up. We're seeing now that our protozoan are coming up, and if you scroll down to the anaerobics, we're seeing a few ciliates show up, but not enough to tell us that we're going totally anaerobic here. We're getting enough watering in the system. 1 ciliate we're not concerned with, like this level of ciliates we're not concerned with. What we are concerned with is a trend over time, right? So if we take this sample and we've got 8,000, and then the next sample we've got 32,000, that's a trend in the wrong direction. At 8,000 and you back it off, or even keep at 8,000, we're fine with that. That tells us we have enough water in the system, but we're just keeping it at that aerobic level. And then you can see that in the battle between fungi and omycetes, we are winning the war now. Now we're starting to select against the omycetes and select for the fungi in the soil. So they're out competing. And then again, spraying the Sufoil product or whatever. It was Sufoil.
It was Sufoil,
yeah.
Which it worked to, I mean, certain times there's a needed necessary action to take. And in that situation, it's a phenomenal product as a knockdown, but it literally kills things by suffocating them. There's a lot of drift. Usually when people are spraying that product, they're trying to kill everything. And so there's a tremendous amount of overspray, or in ag they call it drift, that goes directly onto the soil surface and suffocates all things. I believe between September and October, There was also a change to the instructions of how we instructed to do the extract, and they added nutrients to the extract.
Yes,
And
that's
between
right,
this period
that's
and
right.
this period, where we were trying to make one last boost to pop those organisms up, they essentially nuked the extract before they put it out by adding nutrients, trying to stack functions and do two things at once. Found out the hard way that what they added to the extract turned out to be extremely antimicrobial.
Yeah. So we just show this to you to see that there are a lot of things that when we talk about balance, there's a lot of things that the microbes will tell you in that balance that you can start to understand what you're doing in your cultivation practices and what you're doing in your nutrient and mineral management that can either be helpful or not so helpful in allowing this microbial team to establish. Mm-hmm. So we're going to jump over to greenhouse 6 to see what happened over there. So in greenhouse 6, again, the first soil that we took a sample of was on the same date, August 12th. You can see it's about a similar story. High bacteria, not so high on the fungi, fairly low. This particular soil had adequate nutrient cycling in the flagellates, amoeba, and the bacterial feeding nematodes. We always like to see that in the blue, but there is some adequate nutrient cycling happening there. We are getting a good amount of moisture because we're seeing a few ciliates show up, but not excessive. And then because that fungal component in the soil and the total fungi number is not high enough to compete, the omycetes are able to compete with them. So they're neck and neck on this particular sample. So again, we changed up the brewing practices. We made sure we dialed in their nutrients and their mineral component. They added on all of the mineral doses and made those changes. We did not test this particular greenhouse in between, but we did test it in October, and this was the number in the soil that they were able to maintain the moisture really well with. And so you can see the bacterial populations came right down into a healthy balance at 1,100. The fungal component came right up into the green, so 583. That's a lovely proliferation of the total fungi numbers. Then we're getting really healthy numbers of your protozoan, your protozoan populations with your flagellates, amoeba, and your bacterial feeding nematodes are also there showing up as well. We did get a predatory nematode showing up as well. And then you can see down here that we did reduce in our omycete level, and we have increased in our competition from the beneficial fungi numbers. And so this ratio of omycete to beneficial numbers came way wide. So we're much higher than in our total fungi, which is your beneficial fungi, than we are in our omycetes. We are winning the war on this, and we're creating that pore space that we need to in order to keep it in this zone and select for the guys we want to show up.
I think it's important to note that between this August and October sample in greenhouse 6, remember they were only low on calcium, not also toxic in nitrogen.
Right.
So very quickly, once we got soluble calcium into the system, the mite pressure went away with a couple knockdown sprays, and they actually stopped the 3-time sprays in greenhouse 6.
Yes.
So you can see the difference between greenhouse 1 and 2, where there was not a significant change because the spraying for pests continued.
Because of the high nitrogen?
Because the high nitrogen was driving it. And then once it's very easy to stop the powdery mildew. They made a couple knockdown sprays. What they were using for powdery mildew was not as detrimental to the soil organisms. And so they were able to back off and stop spraying altogether in greenhouse 6. Also, this set of houses was, a couple weeks ahead, so they got to the point where you stop spraying sooner, which is reflected in these organism populations. So they got to the desirable ranges much faster, which had a very clear effect on yield and outcome, quality, potency, like we've gone over in the first two episodes from last weekend, where we covered the difference between potency, bag appeal, and yield.
Yeah. And so this last sample that we have all the way on the right is from the same greenhouse 6, but in the soil that was slightly different than the rest of that, where we've labeled it bad, where they just couldn't get the moisture management in this particular soil because of the way that it was blended and because of the components of this particular soil. We just couldn't get good moisture levels to even hold, which means that your microbes are going to be taking a toll and not being allowed to proliferate, which we see in the numbers here.
Yeah.
Your total fungi, your beneficial fungi cannot proliferate without that moisture, and neither can your flagellates and your amoeba. We did see, bacterial feeding nematode showing up, but again, we've got not really much proliferating, and without that beneficial fungi, we see the omycetes trying to win the war there. So hopefully we've painted a picture for you of how all of these components interplay together to either lead you to pests or out of pests into health. It can often be much more complex than just the microbes themselves, because there's so much within the system that affects them. Your minerals affect them, your nutrition affects them, how much you water affects them. Mulch layer, yes. Can a thick wood chip layer also become problematic, like 6 inches?
Absolutely.
I would say it definitely can.
That was a question that came through back when we were talking about gnats.
Yes.
And it definitely can cause the gnats and downstream pest issues.
Yeah.
This would be something I would agree with. I would like to clarify, I'm not saying super dominant or super excessive in nitrate nitrogen is even that beneficial, but it's more tolerable during the vegetative stages. And this young gentleman, or this C. Ray here, made some corrections and stopped seeing powdery mildew after they ramped up calcium. Fantastic. This is an interesting question about the flood. So how much die-off happens in that flood situation? Is it time-related? What would you say?
Die-off of it's definitely time of suffocation, right? So in this particular situation, they were flooded for a while, so they basically had all of the beneficials die off pretty much, or cyst up on some of the protozoan. And then, when you're that anaerobic for that long, now you have a proliferation of the other set of organisms, the non-beneficial organisms. So now you're completely upside down in that situation.
So I'm not sure what a hemocytometer is. I'm not sure what that is. Maybe break it down a little bit better in the chat. What we do is we use a compound-like microscope.
Yep.
There you go, right there. And we use the methods by Dr. Elaine Ingham for doing a quantitative specific analysis that's quantifying very clearly what's in there.
Yeah, to get these numbers there where we're measuring biomass.
So it looks like Mr. Cub has a microscope and hit a wall trying to count microbes, which is why we're here today.
Yeah, so we were... So that's why I kind of came about introducing the concept that you don't necessarily have to learn how to count and measure biomass per se, but you can start to get used to, as long as you can identify the organisms and you can keep track of how many you have and you know what that scoring looks like if I have 5, if I can identify that I have 5 pieces of beneficial fungi in my soil, is that enough? Is that going to give me enough pore space? Is that going to, give me what I need for my cannabis plants, right? So in the microscope class, that's really more what that's about, is getting used to how much bacteria is enough, how much is really too much, just getting used to training your eye. So that's why I created the score system. So you learn how to identify and you train your eye into just how many am I seeing, is that enough? And you don't have to have a spreadsheet and measure biomass in order to train your eye to do that. So I've tried to make it really simple for you so that you don't hit a wall on looking at, we want you to look at your organisms. We want you to make this real to you so that you can see these trends for yourself, and you can get an idea of what's happening.
And Michael makes a good point. When I was starting to tackle this technique after I became convinced that it was a pathway for me, It was a lot more involved than I anticipated, I'm not going to lie. And, the way I approached it was I approached it like it was my job, so it would become my job, which is just the way I was raised. And so once I learned how to fill out the spreadsheet and I learned how to do the technique, I would sit at the kitchen table, not for necessarily 8 hours a day, but I got up every day and approached it like it was my job and just did repetitions. I also come from a sports background. We went to the batting cages. You need to go to the batting cages and do repetition.
Yeah.
The point of Sarah's class that she's created is to make it accessible and easy.
Yeah.
We've done so many of these biological assessments that we've been able to distill it down into a more qualitative assessment where you're just counting organisms in a certain number of fields of view. And then Sarah's created a scorecard that helps you organize that to put yourself into these orange, red, green, or blue boxes.
Yeah. So you can just trend it over time. How am I trending? Am I seeing more ciliates? How many am I seeing? And are they, if I check next week after I gave this much water, is that now more ciliates or is that less ciliates? Am I, who's waking up and who's not? That's really the idea.
So we got a question about what type of extracting were we doing on this project in our example?
We did not extract with any aeration on this particular project. We actually hand extracted, and you don't have to have aeration you don't have to have a fancy tea brewer in order to do extraction. And in fact, I feel
most of them are problematic.
Yeah, I feel like it's not necessary, and you can actually extract more organisms by hand. That was actually something that Dr. Elaine drilled into me when I was up on their farm, and I had to make an extraction, go count the organisms, make another extraction with a few changes and count the organisms, and start identifying what techniques of extraction actually led to the highest numbers of organisms coming off into the water. So you can get really good at extracting by hand, and you don't even need aeration. And because you're not adding in food with an extract, you don't have to aerate it. You're literally just using that water as a medium to transplant the organisms into your soil.
So we got an interesting question here. Was the increase in fungi from the complete compost addition through the extract, brewer top dress? How did we get this increase on this sample we're talking about?
So it was through we did change out the compost, yes. So it was through a biocomplete compost that had these organisms in it. We did change them from tea brewing to extracts. So we moved them away from possible problematic issues, right? So tea brewings with food sources, making it easy for omycetes to grow, and switched them over to the extract process instead. And we also switched it out to a very simple dispensing method so that it was easy to keep clean, very easy to keep clean, and we, keep the food resource separate.
Yeah, there's some significant issues with the equipment that most people use and what you're taught to use in the living soil. Again, all built by people that have done zero analysis with a microscope. There are some people in the industry that have provided microscope images, but from a scientific basis, their organism identification is wildly incorrect, and that's led the entire community down using really incorrect equipment that literally proliferates disease. So oftentimes what we do is get rid of the tea brewer, move them to a dang 55-gallon drum and a sump pump, and you'll have significantly better results and less powdery mildew.
Yeah. It's not that tea brewers, it's not that they're all bad. You can have a setup that lends itself to easy cleaning, but the key is, there's so many keys to brewing tea. That's why we get rid of teas, because unless you're at the level where you can actually identify correctly and measure the organisms, we don't want you doing tea, because there's so much to manage. You got to keep the tea brewer clean. You got to make sure that you're not overfeeding it. You got to make sure that you're, the temperature, the altitude, the amount of oxygen management, with all those variables contributing to that oxygen management, are crucial to getting a healthy tea at the end result. You can do it, but you won't know you've done it or not done it if you don't have a microscope and aren't measuring it. I can't tell you, we brew tea all the time, and we have brewed, when we're trying out new recipes, or even when we've tried a tried and true recipe, but the temperature changes, which changes the dissolved oxygen in the water. So there's a lot that goes on with tea brewing, which is why we...
People have no idea
Yeah.
how dynamic it is.
Yeah.
Let's get into some of these images and we'll keep answering some questions as we go.
Yeah. Okay, so I just wanted to show you what we're looking at here when we're talking about some of these organisms. So just to orient ourselves to what we're looking at, this is 400x magnification. So this is the 40x objective. This is the objective that you'll do all of your identification of organisms at. So you can see there's a lot of bacteria in this sample. The bacteria are the very simple, round, smooth dots. They can be round, they can be all kinds of sizes. They can be rod-shaped, they can be round rod-shaped. So what we're looking at in this one, all those little things moving and bumbling about, those are actually flagellates. They're a little bit bigger than the bacteria, and they're fairly clear looking, and they've got a long flagella that they're whacking around, which occasionally you can see the flagella coming at, and they're whapping around these bacteria to try to knock them off and eat them. The big guy in the center that's nice and spherical and very, he looks like he's perfectly constructed, that is a perfectly constructed shell of an Arcella amoeba. It's a type of a amoeba. So when I'm focusing up and down, you can't see it right at this moment, but there'll be a round portion of the center of the, right in there. You can see that that's the center of the amoeba there. So. Anyway, so these are the beneficials. This tells us that this is aerobic organisms going on in this soil, and we've got a lot of flagellates moving in and out of the field of view here.
We get a lot of questions about microscopes. We're going to be making a video. The one that's behind us here is
A Meiji.
here. That's a Meiji MT31, which does end up being about a $700 microscope, but it's been absolutely bulletproof for travel. If you're a Soul Food Web consultant that plans to travel to farms, that one was remarkably reliable for 2 years. But we're starting to find some other more affordable ones, and we'll make a dedicated video here coming up in the next week, and we'll just answer all that stuff really completely.
Yeah. Okay, so notice, to orient ourselves to what we're looking at here, this is actually at the 100x total magnification, which is your 10x objective. This level of magnification is not where you do your actual identification of organisms, but I wanted to show you that even at the scanning level, like this is the level that we would be scanning for any nematodes at, you can see this little guy moving around here. And what is he? He looks like he's a race car driver, right? Moving very fast, switching directions really quickly, just moving very, very fast in the soil. This is going to be a ciliate, basically. But in order to actually identify, I mean, I know this because I've been doing this a lot, but in order for you to actually identify it, you would need to rotate into the 40x objective for a 400x total magnification and identify this guy. But the way that he's moving like this, you just don't see any other. This is part of the way that we identify organisms, is by the way that they move. And you'll see this movement in ciliates often. Sometimes they can move slower, but usually they'll move that quickly. And if I were to zoom in on this at 400x, he'd move right out of my field of view. It's so small. So I would capture maybe a little bit, and it'd be very dizzying. So that's why I took it at this level of magnification.
And would this be something that indicates too much watering?
Right. So if you're seeing one ciliate on your slide and that's all you see, I would say that that's not too much, but I would not add more water. So that would be your threshold of water. So, one ciliate on the slide like this would not necessarily be too much. You want to trend this over time to make sure that you're not seeing more and more ciliates showing up. Then you would be too much.
There's a question about King Stropharia that's kind of interesting.
Yeah, do you use King Stropharia? I'm slightly worried around it taking over the top layer and stopping oxygen from reaching further into the soil. Have you gotten? All right, we don't find that. We don't find that that it does suffocate the soil. We find them to be very beneficial. They love breaking down that organic matter. And if you can get that organic matter being broken down by a beneficial fungi like King Stropharia into the soil, now you're getting those nutrients from the organic matter moving down into your soil, feeding the soil life below it. So we don't find that it does suffocate. I wouldn't be worried about that unless,
I'd say that it actually helps because what the King Stropharia does is it actually creates a layer where it actually spreads the mulch layer away from the soil surface. And then one of the ways that the soil goes anaerobic or lends itself to something like the ciliate, which is this guy, is excess water from a microbe standpoint on the soil surface. So it doesn't look like excess water to you, but to the microbe, there's a standing film layer, I guess you could say. So the King Stropharia creates like this spongy surface layer that interfaces between the soil and the mulch layer in your cover crop. And so when you put water into the soil, the King Stropharia helps spreads it out to lessen that excessive water phenomenon. And so I would say that the King Stropharia actually keeps you from going anaerobic. Right. When we make a soil, we keep in mind that there's aeration in the root zone, there's perlite, there's pumice, there's lava rock. We are leading to aeration in the root zone through mechanical means. And people forget that there needs to be aeration and aggregation at the soil surface. Yes. If you just have soil and you stack mulch on top of it, then you're going to suffocate that soil. That's the cover crop lifts up the mulch layer. Mm-hmm. The King Stropharia does a fantastic job of handling that. Mm-hmm. And so I think it actually helps. Another thing that I think the King Stropharia helps do is when you top dress with the minerals, it doesn't allow them to kind of suffocate in big piles and spreads them out, and you're putting them on top of the thing that's taking above ground organic components and turning it into below ground nutrients.
But again, we're not using minerals in the sulfate forms typically, and if we do, it's very, very minimal doses everything when it comes to microbes. So this is a root feeding nematode. If we orient ourselves, this is at 400x magnification where you would actually do your identification at. I want to point out that as he's moving around, and I'm desperately trying to get his head from when he's moving it up and down in the slide,
get it to a pause here.
You'll see that he's got like this spear. Mm-hmm. And when he bends his head, it doesn't go with the bend. It stays stick straight. And at the very end of it, there's a little bulb, just the tiniest little bulb at the end of it. And that indicates to you that is a root-feeding nematode. We don't get all up in arms if I see one root-feeding nematode and I see all of these beneficials in the soil. Where did he come from? He could have come from lots of places. The key is he going to be able to proliferate? Is he going to be able to multiply and cause some serious damage to what's going on? Or does he have competition that's telling us that overall what's happening in the soil is overall in the beneficial zone? Mm-hmm. But you cannot manage what you don't measure. If you're not looking and finding these guys, and over time, then you won't know what's happening. And so I just paused it where she's talking about it. It's like a little needle in there. It's kind of funny. It looks just like the male genitalia, which is pretty funny. It's pretty clear when you see one, and it's fairly small compared to like a bacterial feeding like a bacterial feeding sometimes has a shoot, so it's a little bit longer than that. But you can see how the head is turning, but the spear is not, because it's an actual poker stick. Right. So it stays pretty rigid, I guess you could say. And the reason I point out that bending of the head is because when it comes to identification of your fungal feeders, they don't have a knob, and it can be a little bit more difficult to see the spear, but when they bend their head like that, you can definitely see the spear that doesn't bend with it. So Yeah, so there is a technique for that. We call it flicking the Bic. You're going to need to basically wave a little Bic lighter underneath the slide without turning it black, of course.
Which is kind of tricky.
Which can be tricky to get them to slow down so that you can identify their parts. So this is also at 400x where we do our identification at. This is what we call a rotifer, cute little guy, but he is also an anaerobic indicator. So again, if we see one of these guys in the soil, we're not going to freak out if we have beneficials in the soil, but we are going to freak out, or not freak out, but we are going to understand, we are going to make action. Thank you. Over time, if these guys start proliferating in the soil. He's a cool little dude. You can see him like filtering on the inside. He's got tucked into him, he has these little spinning fingers that he'll pull out when he extends his neck here. But he didn't really extend his little fans this time in this particular picture. But they do have these little fans, and they basically create these vortices that just whir around and suck in the bacteria. And he'll spit out the bacteria he doesn't like, and he'll suck in the bacteria that he does like.
How do you feel about compost extract through the whole plant cycle?
It shouldn't hurt anything. You certainly can do it. I would just make sure that you try not to get any excess of organic matter on top of the soil to suffocate it. So just be mindful to make sure that if you're getting a lot of debris in your extract, that you're not piling that up and causing anything to go suffocation at the top of the surface. So.
Yeah.
What is the best method to make biocomplete compost, and how do you feel about the Johnson-Su bioreactor method? Well, so I'm going to say it depends. The best method is going to depend on you and what you have the bandwidth to actually do. So you can make really great biocomplete compost in a thermal fashion. That will be the fastest, but it's the most amount of work. So you need to be able to be there to assess it morning and night and turn it on time because you're the one managing it. If you have the time to do it and you want your compost to be done faster, then that's what I would go with. Either way, the either thermal or the bioreactor, you still have to suss out your nitrogen component to find out what will actually lend itself to high heat in the pile, and that can be a doozy. Either, excuse me, either way, all this talking. Either way, you are still going to have to do work to make sure that you get a good nitrogen component in the soil or in the compost so that you get the heat that you need. You're going to need that heat in the bioreactor as well. We really like the bioreactor method. Obviously, we have videos on how to do it and how we actually do it, and it's really just a concentrated how to get it done, all the ins and outs in those particular videos if you want to dive into that.
Yeah, there's a question about compost. We sell the compost that we make at cggdistro.com, which I just dropped in the live chat. I guess I can link it in the description.
Yeah.
We've been working with a compost manufacturer since 2016, 2017. I'm pretty proud of that process. And
and I do post the bioassay report of what those numbers look like on our store as well, so that you can see. So, okay, so back to the kind of orienting yourself to what you're seeing, right? Making sense of what's going on in the microscope. So this was a sample when here we're looking at it at 400x total magnification. This is at 1 to 5 dilution, and the reason I'm making this differentiation here, you'll find out, is look at all that we're looking at here. This is a ton of bacteria, but it's not all bacteria. So bacteria are going to be shapes and smooth. So there'll be round, there'll be rods, and there'll be round rods, basically, and they can be in chains and different things like that, but they're not going to be jagged in any way. This particular soil was from a clay soil. Clay can be the same size as bacteria. So you need to be able to know what these differences are when you're looking at it, and you need to be able to say whether or not something is too high or not too high. And so I'm just showing you how you go through kind of analyzing your soil sample here. You can see I take a field of view, I scroll all the way up to the top of the sample, and then I slowly go down through the sample, taking note of the bacteria, taking note of what I'm seeing along the way. A lot of the flagellates will be floating around the top. Here now we're getting down to the bottom of the sample. You can see that we do actually have some fungi in here, that big long strand, but it's not, look how dense it is. Can you really see what's going on? You know, you can see this particular piece of fungi. You can see that we do have bacteria going on, but look how tight it is. You know, if you've got flagellates in this sample, they could be hiding underneath this material. Typically, if they're eating and they're moving their flagella around, they'll be moving these rocks, these pebbles and sand, silts, and clay around, but you won't, you know, if you can't see him to make that identification, bacteria can move things too. So, you know, you don't really know. Mm-hmm. So what I did is I took another sample, which we'll get to here.
Yeah. Will you be teaching the quantitative analysis in the course, or will you be...
I won't be teaching the quantitative. I won't be teaching, not on this particular course. I'm trying to keep it super easy where you're not measuring biomass, but you are taking count, and you do have to do identification of these organisms. And it's just to orient your eye to what you're seeing so that you can get used to taking a quick look at your soil and trending that over time. You won't, you know, because you aren't doing biomass, you won't be able to dial in exactly what your F to B ratios are, but you will be able to say, my bacteria is high, my bacteria is too low, I've got too many ciliates, I need to back off on my watering. You know, it's just to get you looking at these guys so that they become real to you, to trend and make these adjustments in the field, on your watering, things like that. That's what it's about. So you'll have a scoring tool that I'll provide for you, and then you can trend that over time. Mm-hmm. And you'll have the coloring coding too. Mm-hmm.
So here now you've increased your dilution to be able to see the high bacteria.
Yes. So I've taken that same sample, I've diluted it 1 to 10 mils, so it's now still at the same dilution, or same total magnification at 400x, and you can see as I go to the top of the sample here and I start to scroll down through the sample, we're starting to pick out even better what bacteria is there, how much is there, and make a differentiation between the jagged particles and the smooth particles. You know, and as I come down to the bottom of the sample here, you can see there's lots of space. There's a lot more space in between all of these minerals and organic matter. So I can start to see things moving around in here, and I would much better be able to see whether or not I have flagellates that are going to be moving about, or whether or not I do have any amoebas or you know, anything else in the soil that I would need to be able to see. There's not a whole lot going on in this particular sample. We're moving to the next field of view, or at least in that field of view, right? So the next field of view, we're going up to the top of the sample again. We're going to scroll all the way on down through it. You'll get to see what bacteria is there. I'm mentioning the bacteria because it is actually important to identify what bacteria are present. You know, what are the shapes and what are the different sizes? That tells us diversity of bacteria. That tells us whether or not we're increasing in diversity or whether we're decreasing in diversity, right? The robustness of the living soil system. So again, we're now at the bottom of the slide here. We've got a lot of organic matter. We've got a lot of minerals going on, but there's not a whole lot more going on. There's probably there's an actinobacteria going on over in this lower right-hand corner. So you can start to see a few things more in the sample going on than you would be able to in the 1 to 10 or 1 to 5 dilution with this particular type of soil. So it's super important that you learn how to read soil. And so here we are again at another sample, just trying to show differences, right? So this is still at 1 to 5 dilution. If you were learning to count your organisms, this would not be where you make your count at. But at this total magnification and this dilution, this is far different levels of bacteria in this soil than obviously the previous soil, even at the 1 to 10 dilution. So is this too low? Or is this, right at the threshold? Or is this, somewhere in the middle of the green zone, right, the healthy zone? I would say this is probably more around the bottom of the healthy zone, the very minimum amount you'd want to see in your sample. So I'm just going through the top of the sample down to the bottom of the sample, showing you how we move through a slide to really get an idea of what's going on.
And so just to compare again, this is high bacteria at the same dilution, and this is extremely high. What would you say is 10,000 bacteria or something?
Yeah, I mean, this is going to be closer to 5 to 10,000 bacteria. I mean, you'd have to dilute this all the way down to do your counts, probably at least 1 to 1,000, maybe even 1 to 5,000 to do a count on this guy.
So that would be many times higher than desirable.
That's right.
And then this would be at the bottom and maybe not enough.
Yeah, it would be at the very minimal bottom amount of bacteria. You wouldn't want to go much lower than this amount of bacteria, really.
Yeah, that's a pretty low sample altogether.
Yeah. And so obviously this is just 1 or 2 fields of view of each one. You would need to go through many fields of view in order to really get an idea of everything that you're finding in the sample, to really get an idea of whether you're just at minimum, below minimum, or if you're in the good healthy zone. So
yeah, and compost is the solution for everything, in our opinion, really. Fixes most things. So it sounds like Sean's outside of the United States, which we are not able to ship compost outside of the United States. So looks like you're going to have to get onto that Johnson Sioux compost strategy. Here's another interesting question. I've seen that most humic and fulvic nutrients say to add way too much. Do you also see that most nutrient labels are at least twice as strong as what you would really need? And in science, they have this concept called LD50, which is half of the lethal dose. It seems that most nutrient companies use the LD50 as what you should use. To give you some comparison, molasses is a good one to compare, not necessarily talking about humic and fulvic acids, but a lot of people would put, I've seen crazy shit like cup per gallon, so like 240 milliliters per gallon, or even a quarter cup. So now you're talking like 60 milliliters per gallon is a very common dose that people would use for molasses. We've had fantastic results at like 1 to 2 milliliters molasses per gallon. I mean, that actually then accomplishes the goal. So what people don't realize, and what we're trying to just essentially hit you over the top of the head with if you haven't been paying attention, is that most of the things that you're doing are actually extremely antimicrobial, either from a antagonistic, either from a direct killing standpoint, like IPM just kills all the organisms in the soil. Even if you're brewing tea, you're still killing all these organisms you think you're making. If you're following the correct advice or the more effective advice of doing a compost extract with actual biologically complete compost, but you're still spraying IPM 2-3 times a week, you're wasting compost. And on the other side of the scale, if you're putting cups of molasses into your system, you're actually, again, collapsing the soil food web through this concept we're trying to impart of a boom-bust approach. And so any food resource, whether it be fulvic and humic acids, whether it be malted barley, whether it be, nutrients, anything with a stimulating effect on organisms is going to have a threshold to where it's actually very detrimental, even if it's a beneficial compound.
Right.
Yeah.
So dose is everything, and it's dependent on what it is you're talking about. So.
Right on. Thanks for joining us, Mr. Cub, and everybody else that joined us today. Keep in touch. Like we said, we're going to be trying to make a regular deal of at least Wednesdays at 7 p.m. Pacific time. We're going to maintain some of these Saturday, Sunday sessions, but at some point our coursework is going to start here February 5th and February 6th. And so this midday Saturday, Sunday is going to be occupied by our master classes. We will be keeping regular presentations going on at least Wednesdays and hopefully one other regular time per week. We've also got some interviews set up. We're going to get back into our podcasting mode and start having video guests on the show. We're going to be doing some live microscope analysis with Sarah's microscope and all kinds of fun shit coming down the pipe, kiddos. So greetings to New Zealand. Glad you're tuning in. Careful down there where their governments are getting batshit crazy. Stay safe.
Yep.
And
Peace out.
Peace out, homies.